As Johannes Peter Müller posited in 1831, a good physiological experiment, like a good physical one, requires that it should present anywhere, at any time, under identical conditions, the same certain and unequivocal phenomenon that can always be confirmed. Accordingly, a reliable experimental model should fulfill the following criteria. The mechanical force used to induce injury should be controlled, reproducible, and quantifiable. The inflicted injury should be reproducible, quantifiable, and mimic components of human conditions. The injury outcome, measured by morphological, physiological, biochemical, or behavioral parameters, should be related to the mechanical force causing the injury. And the intensity of the mechanical force used to inflict injury should predict the outcome severity. When we translate these requirements to blast injury models, it becomes obvious that modeling blast injuries should reproduce mechanical factors inducing injury based on the physics of field explosions, and the essential hallmarks of blast injuries including position injury severity and injury pattern, graded functional deficits depending on the intensity of the blast, and graded induction of molecular responses as seen in patients. There are several decision-making steps in the process of choosing a model for blast research. Most importantly, the researcher should identify which of the blast effects should be reproduced. If the choice is primary blast, the experimenter should ensure that the animals are restrained so that there will be no blast-induced acceleration of the body or head during the exposure. In a situation where the body or head is allowed to move, the injury mechanisms would involve both primary and tertiary blast effects. This would make the interpretation of the results difficult. Next, a decision should be made about the biological complexity of research study. This will determine the choice of the biological surrogate used to reproduce the blast-induced pathologies seen in humans. For example, researchers will need to consider whether cell culture, tissue, small or large experimental animals, or non-human primates are needed, the positioning of the biological surrogates, the means of generating a shock wave, such as open field, shock or blast tubes, and length of experiment, among other things. Based on the research question and the scale of complexity, a choice is made between non-biological models and biological models. The success of a research study using biological models, especially at the whole animal level, depends on the rigorous selection of animal species used as experimental models. Rodents are the most frequently used experimental animals in trauma research. Their relatively small size and low cost permit repetitive measurements of morphological, biochemical, cellular, and behavioral parameters that require relatively large numbers of animals. Because of ethical, technical, and/or financial limitations, this is less achievable in phylogenetically higher species. Nevertheless, the anatomical and physiological differences between humans and rodents, especially in circulatory and nervous systems, limit the utilization of small experimental animals in blast injury research. Experimental studies on primary blast-induced biological effects routinely use either shock tubes, which use compressed air or gas to generate a shock wave, or blast tubes, which use explosive charges to generate a shock wave. For example, in a mouse blast injury model built by the Johns Hopkins University Applied Physics Laboratory in Laurel, Maryland, a compressed, helium-driven shock tube was used. The 14.6 or 6-inch diameter pneumatic shock tube system consisted of both a driver or pressurized and a driven or ambient section. Before each test, a diaphragm was inserted between the two sections, creating a closed volume driver section that would allow pressurization. The driver section, which initially contained ambient air, was pressurized with helium gas until the pressure differential between the driver and driven sections exceeded the material failure threshold of the diaphragm, thus initiating rupture. The diaphragm rupture pressure levels can be varied by selecting various combinations of Kapton and polyethylene sheets. Upon diaphragm rupture, a shock wave propagated down the length of the shock tube. Side-mounted pressure sensors, used to record the static pressure and wave propagation, were installed at 5 locations along the shock tube inner wall. Anesthetized animals are fixed individually in special holders designed to prevent any movement of their bodies in response to the blast, and thus to prevent tertiary effects of the shock or blast wave. The single driver shock tube system generated dynamic overpressure loading conditions necessary to induce mild, moderate, and severe levels of lethality in mice. The main challenge in researching complex pathological conditions, such as blast-induced neurotrauma, is to develop experimental models that reliably mimic human conditions and manifestations of the disease. It has been reported that immediately after a blast exposure, soldiers have experienced memory loss for events before and after the explosion, as well as confusion, an impaired sense of reality, and reduced decision-making ability. Later, sometimes months and years after blast exposure, patients with blast-induced neurotrauma have reported irritability, speech problems such as reduced verbal fluency, impaired working memory and executive functioning, headache, dizziness and balance problems, as well as psychological impairments such as depression and post-traumatic stress disorder. Cerebral arterial spasm and intracranial hypertension are frequent and significant complications of moderate to severe combat blast traumatic brain injury. Vasospasm can develop early, often within 48 hours of injury, and can also manifest later, typically between 10 to 14 days post-exposure. Moreover, a lower relative cerebral metabolic rate of glucose has been observed in veterans 15 to 86 months after blast, as well as cerebrocerebellar hypometabolism 2 to 5 years post-blast. Comparable functional deficits have been observed in animal models of blast injuries and blast-induced neurotrauma. This video shows a test of spontaneous exploratory behavior after blast exposure, assessed using the open field test. The test was conducted in a white, opaque rectangular polypropylene arena measuring 45 centimeters by 45 centimeters by 40 centimeters with a lamp positioned 30 centimeters above the arena and providing 550 lux illumination. Each mouse was placed individually into the center of the arena and allowed to roam freely in the box. The animal's movement was recorded over a 5-minute period by using a video camera on 7 consecutive days before injury, and on days 1, 2, 3, 4, 5, 7, 10, 14, 21, and 30 days post-exposure. The tracking program AnyMaze was used to calculate both the frequency and duration of a range of behavior. The parameters included distance covered, mean speed of movement, number of rearing instances as a measure of exploratory activity, number of center entries, time spent in the center, and time spent immobile. The clip shows the movement of a healthy mouse, which covered the entire surface of the field, showing curiosity and interest toward both the central and peripheral fields as well as the corners. This open field test shows the movement pattern of a mouse exposed to a mild intensity blast generated using a shock tube 1 month after the injury. The mouse shows reduced spontaneous activity with a preference for the peripheral field and corners. The freezing behavior, which is manifested by seeking shelter in the corners, a lack of interest in the animal's environment, and the avoidance of any activity, is consistent with increased stress and anxiety and is not associated with decreased sensory or motor deficits. Such a response in animals has been compared to post-traumatic depression in humans. The motor function was tested on days 1, 2, 3, 4, 5, 7, 10, 14, 21, and 30 after blast exposure using the rotor-rod test, which has been described as being one of the most sensitive tests to detect motor deficits in rodent brain injury. Briefly, animals were placed on a rotor-rod device. For training, the mice were allowed to explore the rotor-rod for 2 minutes without rotation, and then the drum was slowly accelerated to 14 rotations per minute. The mice were subjected to 2-minute training trials for 7 consecutive days before exposure. For motor function tests, the rotor-rod test was performed first by placing the mice on the drum rotating with 25 RPMs for 2 minutes, and measuring the length of time each animal was able to maintain its balance walking on the top of the drum without falling down. This video clip shows a rotorod test performed with a healthy control mouse positioned on the left side, a mouse exposed to mild intensity shock wave 1 month earlier positioned on the right side, and a mouse exposed to moderate intensity blast wave 1 month earlier positioned in the middle. All animals exposed to blast demonstrated a significant decline in motor performance. These motor deficits were observed during the entire 30-day observation period. The animal with moderate blast injury in the middle demonstrated the most prominent changes.
The cerebellum is part of a frontocerebellar system that, when disrupted, results in significant cognitive and behavioral consequences. Mebon and colleagues found in 2016 that the number of blast exposures correlated with the uptake of FDG, a commonly used indicator of neuronal activity in the cerebellum of veterans. Furthermore, in experiments with a mouse model of blast traumatic brain injury, they showed that blast exposure produced microlesions in the blood-brain barrier, predominantly in the ventral cerebellum. Purkinje cells associated with these BBB microlesions displayed plasma membrane disruptions and aberrant expression of phosphorylated tau protein. Purkinje cell loss was most pronounced in the ventral cerebellar lobules, suggesting that early-stage breakdown of BBB integrity may be an important factor driving long-term brain changes. Blast exposure caused reactive gliosis in mouse cerebellum, particularly in the deep cerebellar nuclei. In comparison, diffusion tensor imaging tractography of the cerebellum of blast-exposed veterans revealed that mean diffusivity correlated negatively with the number of blast-related mild TBIs. Together, these results indicate that the cerebellum is vulnerable to repetitive mild TBI in both mice and humans. In our experiments, we subjected the mice to mild intensity blasts generated in a shock tube and assessed neurological function and behavior using the open field test paradigm. Persons with an ataxic gait due to cerebellar dysfunction keep their legs further apart than normal, a position referred to clinically as broadened base. They often stagger and resemble persons who have ingested excessive alcohol. Moonwalking, made famous by Michael Jackson, is a dance move wherein the actor appears to walk forward but is actually sliding backwards. The mice seem to do it because they place their feet further apart than normal as they walk in order to maintain their balance. 7 days after being exposed to sub-minimal blast exposures, this mouse demonstrates the typical moonwalking phenomenon. That is, the mouse walks backward while its intent is to move ahead. A similar moving pattern has been seen in a mouse model of dominantly inherited cerebellar ataxia, a so-called moonwalker mouse, that demonstrates abnormal Purkinje cell development and early changes in Purkinje cell physiology. Such similarity supports the vulnerability of cerebellum towards the effects of blast-induced pressure wave.